Welcome back friends. Welcome to another video from Shomu's Biology. And in this video tutorial I'll be talking about recombinant DNA technology. We have talked about many different aspects of molecular cloning and genetic cloning, but we didn't talk much about recombinant DNA technology in a whole. So we'll be talking about recombinant DNA technology in maximum amount. So let's begin with it. In short form we write it as a RDT. So there will be series of videos regarding recombinant DNA technology. Now this is the first video of recombinant DNA technology, and here we will be talking about the cloning. That is the most important thing in recombinant DNA technology. The cloning, we call it a DNA cloning, we call it a gene cloning, whatever you can say. So it's a DNA or gene cloning, or you can also say it as a molecular cloning. So what is cloning? So I'm going to give you a big picture, an overview of whole process of molecular cloning. Okay, so it has several different stages. Okay, so at the very beginning, what is this molecular cloning? You probably heard this term clone or cloning in your life. Now you also know about that it is duplicating something, not actually duplicating but also producing of same copy of something. That means if you have a, say, a mobile phone, right, and you can make same type of mobile phones again and again and again using the same structure and features and all the stuff, it will be called as a cloning. So a particular model number of a mobile phone, of a smartphone, there are multiple copies out there, so those are the clones of the same type of the mobile. Okay, so there are multiple examples of such cloning thing, but here the cloning is for the DNA cloning or gene cloning. So we are talking about cloning, that means creating multiple copies, multiple same copies of the same DNA and gene again and again. Okay, using bacteria as a vector. Okay, not vector, actually bacteria as a host organism, and using things called vector, we'll be talking about later. But the thing is, we have a thing, we have a gene, we have a DNA, we need to make multiple copies of that. That is the idea. And the idea is very, very simple, right? So for this cloning to happen properly, what we require, at least the gene to be cloned. The gene or DNA to be cloned. This we require. Okay. Now for example, let's assume here, for example, the gene to be cloned is a human gene. Say it's insulin. This is the gene. This is a human insulin. Okay. Human insulin producing gene. That is the gene we want to clone. Now the thing about cloning is that what we want to do, we want to amplify the number of the same gene so that we can produce more amount of insulin in this case. In other cases we can produce more amount of other proteins or other factors, whatever we require. In this case we require to produce a lot of insulin for commercial purpose, for our requirements, because insulin hormone is required for diabetes disease. So in that case we will do that. This is the thing, this is the first thing you require. The second thing you require is the process, the whole system of amplifying the same type of gene again and again so that they can produce into proteins. Now how can you do that? Now you may tell me one thing, that the gene we can amplify with PCR or polymerase chain reaction, right? So why we require to do a cloning? Now the thing is, polymerase chain reaction is an artificial process where we amplify only the segment of DNA in a test tube, okay, or in a Eppendorf tube, whatever, PCR tube. Those are in an artificial tube or artificial chamber. We amplify them. But those DNA or genes, whatever we produce, those will be fragments of the DNA or fragments of the genes, the exact small amount of the gene only. Let's say the small fragment of the gene only. Now that gene is not enough to produce protein, because they cannot produce protein on their own. For proper production of proteins, what else do you require? We require a start point of protein synthesis, a stop codon of protein synthesis right above and upstream and downstream of that gene for making the protein. So if we can amplify this gene with PCR, but that's not enough to apply it to produce proteins, because ultimately what we want proteins. That's why we are cloning things most of the cases. Okay, we want to know what's going on there. That's why you clone these things most of the cases. Okay, so in those cases PCR amplification is not the answer for our question. So we want to do it in a way so that we can put it in a host cell and it can produce some proteins out of it. So that's why we need a separate approach. What is that approach? We take that gene, in this case human insulin, this is the gene, we take it from human chromosome. Then what will you do? We want to amplify it. So for the amplification, we need to insert this gene definitely in such a way so that it can produce multiple copies of itself. So insertion of the target gene in a host cell, because you know inside the host cell DNA replication is going on all the time, transcription is going on, translation is going on. So we want this gene to be inserted in such a host cell. Let's say in this case it's a bacteria E. coli. This is a bacteria. Inside this bacteria we want to insert that host gene so that they can be amplified naturally inside the host cell. This is the second process. And once it is amplified, then what we need to do, the third thing, to extract the DNA or product, whatever we want, because sometimes we want to insert into the host and make proteins directly. Then we extract the product as a protein. So these are the three major stages. Okay, the major part in an overview, if you look, you need to have a gene, you need to insert it into a host, you amplify, then you need to take out the products. These are the three linear stages if you look from outside, big picture. Okay, but now the detailed process of all this thing. So what we begin with, you know, it's very easy to obtain the gene of interest. So you have human genome. Let's say this is a huge human genome. Among this genome there is a small portion, this red colored region, this is the insulin gene, and we know which chromosome contains that gene and where exactly this gene is, and we need to take this gene out. So we can cut this gene out. This is the first step. We need to cut gene out, the desired gene. Now the second stage is the amplification. That means we need to insert it into the host DNA. E. coli has its own DNA and its plasmid and all this stuff, but we need to insert this cut fragment of DNA inside the host. So how can you do that? You cannot directly just, because if we cut this DNA, this is a segment of insulin DNA for example. How it's done, we haven't talked about yet. We'll be talking about it later. But this is cut. Then what we need to do, we need to insert it into the host DNA, host body. So this is bacteria. We need to insert it into the bacteria. Okay, but how? If we just simply take this cut out of the DNA, insert it into the bacteria, this part will be functionless, because this gene will only function if it has an origin of replication, because we want this DNA to be cloned, to be replicated, to be multiplied. So we require an origin of replication upstream of the gene for making this gene multiple copies, right? So we cannot directly put it like this. So we need a vehicle with which we will insert this DNA to the bacterial cell. That we require, right? This is another important stage, and we call that thing vector. The vehicle with which we insert our target gene into the host cell during cloning is known as the vector. Okay, we use vector to insert it. Okay, how and what is the whole process? Still not talked, we'll be talking later. But now once we know, using the vector is nothing but a genetic segment. Most of the time it's plasmid, it can be different variations of plasmid, but for example, for now on, just think of as a plasmid. It's a portion of DNA which is self-replicating. which has the capability of replicating itself. So we have this plasmid and we have this gene attached. This is now inserted into the host cell. Once it's inserted to the host cell, now the stage, it will replicate. So now the whole copy will be replicated inside the host cell. Okay, once it's replicated, then what we need to do, we need to extract out the product, either the gene segments or the products, right? For example, we want them to translate all the proteins, to translate and synthesize the insulin protein. Then we extract the insulin protein by cracking open the E. coli cell and we get the product. So that is the final stage to get the product. This in a sense is the idea of cloning. Okay, now for this idea, remember the important stages from molecular perspective comes here at this point. This is the first point of the molecular perspective. Okay, that how to cut the target gene of our interest. One thing, this is the thing that we haven't talked about. This is, say, number one we'll be talking here. Second thing, we need to insert it into the vector. How we insert it into the vector and what is vector in the first place, we need to know that thing also. So this is the second thing we want to know, what is vector and how we put this gene inside the vector. And then third thing, after putting this gene inside the vector, we allow this bacterial cell to take up that DNA inside. So how can you be sure that the bacterial cell uptake that DNA properly or not? That is called screening of the cells, that which cell took the DNA, which cell did not. That is the screening, third thing. So these are the three major problems with a molecular cloning. How to prepare the DNA to be cloned. See here, how to prepare the DNA to be cloned, how to insert that DNA inside the vector to make it ready to be delivered into the host cell. Third thing, how do you know that the host cell took up the DNA? These are the three big problems with molecular cloning, and we will encounter each of those problems in our detailed videos later. But this is a big picture of how we do the cloning. Okay, once you know this, then rest of the things become easier. Now I'm going to give a little bit insight in this video, then we'll go to the next video, talk about vectors in details. We'll be talking about screening of the libraries in detail. We'll be talking about the different types of vectors that we use and what are the applications of this recombinant DNA technology. Okay, so in this video I'll be talking a little bit more about the detailing of the processes. So here let me erase the stuff here. Okay, so the idea is very simple. We have the target DNA. Now let's say the target DNA is very big. This is the whole target DNA. And this is the whole genome of human being, or for example, this is the target DNA, the red thing. This is the target DNA. It could be insulin or any kind of protein gene. So this is the target. The first stage, we need to cut this target. Second, we need to insert the target and merge the target with vector. Okay, we know vector is a delivery system. It is also a genetic element capable of self-replication. Now take example as a plasmid. We say vector. Vector is a plasmid, right? So let's draw it. This is the plasmid. Circular, you know, plasmid is a circular DNA capable of self-replication. It is frequently found in bacterial cell. This is plasmid. Now, the complete idea. There is a specific, so the idea here is to take this target segment, the red segment, and put it into the vector. Okay, how can you do that? The easy way to do this is we use an enzyme which can cleave the DNA at a specific point only, and we call that restriction endonuclease or restriction enzymes, okay, known as RE, restriction endonuclease enzyme. Endonuclease means those enzymes can cleave nucleotides from the middle, right? Nucleic acids from the middle. Restriction endonuclease means they are restricted to cleave at a specific point only. Usually they have a specific recognition sequence pattern where exactly they will cleave, and those patterns are mostly of a palindromic sequence. For example, if I give you one example of the enzyme, it's called EcoR1. Eco, why? Because it's extracted from E. coli R1. It's depending upon the strain what used to produce this enzyme. So EcoR1 is a restriction endonuclease which detects and responses to the sequence GAATTC. And whenever it finds this sequence GAATTC, this is called a palindromic sequence. Why? Because if you read it from front and back, the sequence will be the same. GAATTC opposite GAATTC. That's why it's called a palindrome. Now, most and all the restriction enzymes that are available, they detect a specific palindromic sequence and they cleave at a specific point. Now, EcoR1 recognizes this palindromic sequence but cleaves right after this GA from here as well as from here. So it will cleave the DNA like this fashion, okay, both the ends. From this second strand, it will cut from this side of GA, from upper strand it cuts from that side of GA. That's how it cut. So it's an example of endonuclease. So this endonuclease, this restriction endonuclease will be applied to cleave this segment out. Now it depends what kind of segment we find, right, overlapping to the target gene. Okay, depending upon that we change the type of endonuclease that we require. So we cleave that DNA. So we cleave this DNA. Now what we have the target DNA in our hand, right, and it is cleaved with, say, EcoR1. Now what we need to do, we need to treat the vector with the same restriction enzyme. Do you know why? Because you know here they cleave in a specific pattern, GAATTC right after GA. So it generates a specific type of cut end. So we need to treat the vector with the same enzyme to create the same complementary cut end so that we can join these two DNA together. If we apply two different restriction enzymes there, EcoR1 for this host DNA to cut, and here say another example BAMH1, different restriction enzyme. In that case, the ends that will generate will not match with each other. So the target DNA cannot be joined with the vector. So for joining the target DNA with the vector properly, we need to cut both of them with the same type of restriction endonuclease enzyme. So you treat it in both the ends. So it also cuts here, a segment out. It cleaves the segment. So after cleaving the segment, we will insert this target DNA to the specific place. Here, for attaching the target DNA to this specific point of the vector, we cleave it with restriction enzyme, we know that. Now how these two things will join? How to join two DNA segments? There is an enzyme to join DNAs also. Example, the name is ligase. You probably heard this name before. DNA ligase is an enzyme which can seal the nick, right? Ligase cannot add any nucleotides, but it can seal the nick, right? So here we use ligase to join the nicks. Nicks are joined like a packet of glue. So molecular scissors are the restriction enzymes and ligase is a molecular glue that seal the nick. So now what we have a construct of vector carrying a copy of our target DNA. This construct is known as recombinant DNA. Recombinant DNA. This is called recombinant DNA or recombinant DNA plasmid now. This is very, very important. That's why the whole process and technology is known as recombinant DNA technology, because here we prepare this DNA with recombination, recombining both vector as well as our target segment. Okay, that is the idea of preparing recombinant DNA. Once we prepare this DNA, then we will insert this recombinant DNA into the host. How we do that? The host, let's say here we take bacterial cell as a host. Say this is the E. coli cell. Okay, so E. coli has a specific structure out there and all the stuff. Now we want E. coli to take this plasmid inside. There are many ways to deliver a plasmid inside. We can use a vector like plasmid. If you use a vector like plasmid, we need to use a process called bacterial transformation for transferring the vector into the host, right? Transformation means we simply apply a chemical, calcium chloride, which make E. coli cell very crazy to take out all the vectors inside, because this is a natural process of transformation, because calcium chloride influence a situation called competence in E. coli cells. So E. coli cells will generate the competence and will start taking up DNA fragments from outside, and that's a natural process in E. coli. Natural ways how bacteria transfer their genetic segments from each other, between each other. That's why we exploit that system, but we simply influence the system by applying calcium chloride, right? And it will uptake the DNA. Now the thing is, we put lot of vector, we make many vectors like this and we put it into the mixture. We call it a ligation mixture, where all this vector DNA is ligated with the target DNA. Now in that mix we also add some, we take some part of this vector and we add it into the E. coli culture. It's filled with E. coli. So now what we know, many E. colis are there, many vectors are there. We apply that competence thing there for the E. coli to uptake. Now the thing is, not all the cells can uptake this vector at that time. It's not possible. Only few cells will uptake it, few will not be able to uptake it. Okay, so then after this whole thing is done, what we need to do, we need to make sure which cells actually took the vector DNA, the chimeric vector or the recombinant vector DNA, and which cell did not take the recombinant vector. That is very, very important. I told you that we need to know whether our cell, whether our host cell contains our target DNA or not, right? So that is very, very important. And how can you know that? Because after this uptake thing, we allow some time for the cell to take this DNA, but then we don't know, we cannot see the things going on. So we need to take the bacterial cell, we need to plate them in the agar plate, the Petri plate, with all these things, with antibiotic resistance, and using antibiotic resistance as a marker, we select which cell took the DNA, which cell did not. That is the idea. That is called screening of clone libraries or screening of gene libraries. Because you know, library, why we call library? Because there are multiple cells and vectors that are taking the things, but we make sure that only we make sure to take those cells only which have our DNA, right? Not the other things. So let's for assume that in this case they uptake our DNA, right? Our vector means vector with the DNA, is the recombinant vector, recombinant plasmid. So it's inside the bacterial cell. Then the cell, what it will do? The number of plasmid copy will increase. How? Normally plasmid, if you use plasmid do not have multiple copies in E. coli cell. Generally it has low copy number there. So the idea is the bacteria will divide. From one cell it will become two cell. So when it becomes two cell, two plasmids, again 2 to 4 cell, 4 to 8, 8 to 16, 16. So it will keep on multiplying like with binary vision, and the target DNA of us also start multiplying with it, because each time the cell multiplies, it also replicates a copy of its plasmid for its daughter cell. And that's how the whole thing works. And soon we are going to get a lot of number of these recombinant plasmids, this recombinant DNA in the mixture. And when you get that huge number of recombinant DNA in the mixture, then we can take those DNA out, we can produce proteins with it, and we can extract our DNA. We can extract our product from that. So this is the idea. This whole process I've told you about, this insertion thing, this is transformation. Transformation, the process of uptake of DNA by bacteria. But there are other processes depending upon the type of vector we choose. If we choose a virus, a bacteriophage virus as a vector, we can choose that. There is a virus called lambdaphage. We can take lambdaphage as a vector. If we take lambdaphage as a vector, we don't need to use transformation then, because that phage can infect bacteria. By that way, automatically the recombinant plasmid or the recombinant DNA will be inserted. There are multiple techniques depending upon the type of vector. But ultimately, that's how we need to transform. It will replicate, and then finally we get our product. So this, in a sense, is a recombinant DNA technology basic. I mean, among recombinant DNA technology, the basics of cloning. And in recombinant DNA technology, we talk about tweaking cloning in different regions, all the small details of this process, the type of restriction enzymes to use, what type of restriction enzymes to use, when not to use. Then we need to also talk about vectors, what are the different types, how to exactly use the vectors, and many more things that we need to talk about. Now in this case, we just see the basics, the overview, the big picture, because it is very, very important to clear your big picture, right? So once you clear your big picture, then the rest of the things become very easy to understand, right? So this is the first video of recombinant DNA technology regarding gene cloning or DNA cloning or molecular cloning, whatever you like to say. And in the next video we'll be talking about in details about vectors, as well as we'll be talking about the different stages and screening process and multiple much more detailed things, and also the applications of recombinant DNA technology. So that's it guys, I hope this video helps you. If it helps you, definitely hit the like button, subscribe to my channel. You probably have this subscribe button here, subscribe, click this button to subscribe to get more and more videos like that every day, and also share this video with your friends. Thank you.